Hello everyone, my name is Breanna Giles and I am a PhD candidate at Deakin University in Australia. Thank you for taking this opportunity to view my poster virtually at the 11th International Aptamers Conference. In this presentation, I will be discussing the potential aptamer-drug conjugates have at becoming drug delivery vehicles for transporting therapeutic payloads to cancerous cells, in particular for glioblastoma. Glioblastoma is the most aggressive form of brain cancer with fewer than 5% of patients surviving greater than 5 years post-treatment. The current treatment options of surgery, radiation therapy and chemotherapy are often unsuccessful, resulting in a strong chance of tumor recurrence. Furthermore, the highly restrictive nature of the blood-brain barrier limits the ability for treatments to reach tumors, thus outlining the importance of developing a novel therapeutic. One such alternative includes the use of aptamers, such as the TEP bifunctional candidate shown here, as a drug delivery vehicle to specifically deliver cancer drugs that cannot naturally cross the blood-brain barrier, including doxorubicin, into cancerous cells. We have previously shown the TEP-DOX aptamer-drug conjugate to be successful at crossing both an in vitro and in vivo blood-brain barrier model to treat brain metastases, so it is hoped that the same conclusion can be achieved for glioblastoma. Firstly, we assess the binding affinity of the TEP and TEM bifunctional aptamers towards the T98G glioblastoma, MDA-MB231 triple-negative breast cancer, and HEC293T healthy embryonic kidney cells. We can see based on the generation of saturation curves and the dissociation constant shown in the table that a moderate binding affinity for TEP was achieved towards the T98G and MDA-MB231 cells, while demonstrating no binding towards the healthy HEC293T cells. As anticipated, the scrambled bifunctional aptamer 10 showed no binding towards either cell lines. However, upon intercalation of doxorubicin into the aptamer stem regions, it is postulated that the 3D structure of aptamers is altered, allowing for 10 to bind towards the T98G transferrin receptor positive glioblastoma cells. This phenomenon was also observed by McDonald and colleagues, who saw a moderate binding affinity towards the transferrin receptor of BEN3 mouse brain endothelial cells, while still remaining nonspecific to the MDA-MB231 and HEC293T cells. We can also see that this altered the binding of the TEP bifunctional aptamer towards the cancerous cell lines, however, it still maintained specificity. Next, we grew T98G cells in non-adherent conditions to develop glioblastoma tumors and assess the internalization of TEP-DOX. With the strong presence of co-localized aptamer-DOX shown here in yellow on the right, we can see that following 2 hours, TEP-DOX has successfully begun to bind and internalize into tumor spheres. By performing an MTS assay to determine the cell viability of T98G and MDA-MB231 cells based on their ability to metabolize their MTS substrate post-treatment, we can see that doxorubicin is capable of inducing cytotoxic effects, especially after 48 hours based on this computed model. It is important to note, however, that while no effect was reported at 24 hours using the statistical program GraphPad PRISM, you can still see that there is a downwards trend of cell viability representing cell death. This trend was further observed with the treatment of TEP-DOX and TEN-DOX, which showed similar half inhibitory concentrations to free-DOX alone, despite a postulated initial delay in drug release following internalization. These results show promise in that the aptamers bring forth a targeted delivery approach for doxorubicin, and based on previous findings by McDonald and colleagues, has the potential to cross the blood-brain barrier to treat glioblastoma. To further assess the efficacy of both aptamer-DOX conjugates towards inducing cytotoxicity in tumor spheres, the T98G cells were grown in non-adherent conditions for 5 days. Upon treating with 1 to 20 micromolar equivalent TEP-DOX and TEN-DOX concentrations for 72 hours, a trackband blue exclusion assay was performed to count the number of live and dead cells based on the cell's stained color of either white or blue, respectively. Here, the preliminary findings further demonstrated that both aptamer-DOX conjugates are effective at delivering drug payloads into glioblastoma tumor spheres, with slightly better IC50 concentrations reported than free-DOX alone. To further identify if the aptamer-DOX conjugates remain specific to just cancerous cells and do not harm the healthy cells, all cytotoxic testing will be repeated with the HEC293T cell line. Finally, I would like to acknowledge Deakin University, the Australian Government and Impact for funding this project. Also a special thank you for all the guidance and support Associate Professor Sarah Shigdar, Dr. Rasika Samrazinga and Dr. Mariam Nakjivani have provided. Please feel free to contact me with any questions at the email address provided below.